I want to start with a brief announcement, and I'm doing this, Brent Smith and Nora Lahr, who is Dan Atwater's wife, are doing a wellness course in trail building and invasive species control of Earlham's back campus and other properties. So if you need a wellness and you would like to do something biologically relevant with the wellness that you need to do, here's an option for you. This is a spring course. It's in the second 7 weeks, and you get your wellness credit and learn some biology from Brent and Nora at the same time. Okay, so let's see. So what we're gonna focus on today is water homeostasis, how the kidney helps us conserve water. Obviously, if we were to excrete anywhere near the amount of water that we filter, we would become dehydrated in a matter of minutes. You learned that from the problem set that it takes about a half an hour for your kidneys to filter a volume of plasma equivalent to the entire body's plasma. That's not exactly accurate because, of course, there's a mixing problem, but overall, you're filtering all of your plasma in something like 30 minutes. So we obviously have to recover a lot of that water, 99% of that water. And that's what we're going to be talking about today. I don't have your problem set because I haven't finished grading them. I realize that one of the questions, my answer kind of depended on the material from today, which you don't have, so I won't look for that in your answers when I'm grading it. Okay, so water. Where do we get our water from? Well, we drink about 1.2 liters a day. We get about a liter of water from food, from solid food, because food has water in it mostly. We actually produce water from metabolism. Remember that oxygen is the final electron acceptor in the electron transport chain. When oxygen is reduced, when oxygen is reduced, it produces water. And so you actually make some water from from the end product of metabolism. So that's what this 300 mils is. There are, just to blow your mind, there are desert rodents, desert rats, that actually never drink water. So their fluid intake is zero. They get some of their water from the moisture that's in the seeds and things that they eat, and they produce most of the water that they need from metabolism. So that's incredible. And you can imagine that those animals have an amazing ability to conserve water, right? So if you're making your own water and you're not drinking any water, you probably have to, your kidneys have to make sure that they don't excrete too much of it, and those rats excrete almost crystalline sodium chloride. I mean, it's a highly concentrated urine, and we'll talk about that later today. So we get we take in about 2.5 liters of water, plus or minus, and then what are the outputs? Well, we produce about 1.5 liters per day of urine. 100 mils of water are in feces. We sweat, we evaporate 500 mils of water from sweating. And we exhale about 350 mils of water just by evaporation from breathing. So that's where, that's on the big picture, that's where water comes and goes. So we produce, the main point here, the main point of the whole class is how do we produce either a dilute or a concentrated urine, depending on the metabolic, the homeostatic needs of the body. So here are some data that were collected in a A&P lab a few years ago. We could do this in lab today, but we're not going to, because we have better things to do. But here's a plot of urine osmolarity, so the solute concentration of urine, as a function of time after drinking a half a liter of water. So I asked the students to come to lab relatively dehydrated. I asked them to not drink anything at lunch and to not drink anything for an hour before lunch. So I asked them to come to lab having not drunk anything for about 2 hours, because lab started at 1. And you can see some were compliant, and they were producing a concentrated urine, and some were not. they were producing a dilute urine. And I could tell who was compliant and who wasn't, but it didn't matter because it just shows the range. If you stop drinking for a couple of hours, you need to conserve water, and the urine that you produce is highly concentrated, 1,000 to 1,200 milliosmoles per liter. Remember, osmole is just the concentration of all solids: sodium, potassium, chloride, phosphate, urea. All the solutes contribute to osmolarity. So when they came in, many of them were producing a concentrated urine, and then within an hour after drinking 500 mils of water, they were all producing a dilute urine of 200 milliosmol or less, sometimes down to about 60 milliosmoles. And so this is, one person went from producing 1,200 milliosmoles per liter urine to producing 150 milliosmoles per liter after drinking a large volume of water. And then we also did an experiment with drinking half a liter of coffee and half a liter of salt water and not drinking at all. And you can see here was someone who came in compliant and they stayed relatively dehydrated, and here's someone who wasn't compliant at the beginning, but an hour and a half later was producing a fairly concentrated urine. So the point is we produce either concentrated or a dilute urine, depending on whether we are dehydrated or fully hydrated. So here are the mechanistic themes that we're going to go through. So I'm going to do this kind of more than once. I'm going to tell the story in, 2 or 3 different ways. So the themes are that when the kidney needs to generate a concentrated urine because of relative dehydration, it uses the loop of Henle to build a hypertonic renal interstitium. What is hyper? Let's unpack those words. What does hypertonic mean?
Water's outside of the cell. The fluid is more outside of the cell.
Nope, not more outside. Well, maybe you're getting it. Hypertonic means, somebody maybe elaborate on that a little bit.
It's a high solute concentration, so water can flow.
It's a high solute concentration. I don't know if that's what you meant, but a high solute concentration is hypertonic. Isotonic means that it's got the same solute concentration, and hypotonic means it's got a low solute concentration. And so the question is, high, low, or equal to what? The what that we're comparing to is normal osmolarity, which is about 290 milliosmoles. That's normal plasma osmolarity, 300. You can round it off to 300. So hypertonic is a solute concentration higher than 300 milliosmoles per liter. Hypotonic is a solute concentration lower than 300 milliosmoles per liter. So you can see here, these people were producing a hypertonic urine, and so is this person, because that's slightly hypertonic. This person is producing a nearly isotonic urine. Here, they're all producing a relatively hypotonic urine. Hyper, high concentration, hypo, low concentration. Okay, just getting the language out. So hypertonic, a high concentration of solute in the interstitium, in the spaces outside, as Zoe said, of the renal tubules and outside of the vasa recta. In the interstitial spaces between the loops of Henle and the vasorectum. Okay? So the loop of Henle is going to build a hypertonic renal interstitium, and I'm going to show you how that works. And then water is going to follow, kind of as Zoe said, water is going to then follow the concentration by osmosis from the urine, or from the urinary fluid, into the interstitium, and then into the vasorectum, where it can be recovered. And you'll see that the water permeability, the permeability to water is going to be key here, and that's going to be controlled at the very last step in the collecting tubule. In fact, in the medullary collecting tubule. Those are the themes. We're going to talk about how we build a hypertonic renal interstitium, where the osmosis tells the water to go, and then how we control water permeability. So now, let's tell the same stories from a slightly different perspective. Tubular fluid starts, well, remember, tubular fluid starts as a filtrated plasma, so it starts isotonic, and it stays isotonic in the proximal tubule, because the proximal tubule is so leaky that when salt is reabsorbed by the mechanisms that you told me about on the problem set, water is also reabsorbed in parallel, and so the fluid stays isotonic throughout the proximal tubule. At the end of the loop of Henle, which was building the hypertonic renal interstitium, the fluid inside the tubules at the end of the loop of Henle is hypotonic, because all those salts have been pumped out into the renal interstitium, and so what's left in the tubule is hypotonic. Okay? It's going to stay hypotonic in the distal tubule, and then, after controlling water permeability in the collecting tubule, it either remains hypotonic if we need to produce a dilute urine, or it becomes hypertonic if we need to produce a concentrated urine in the collecting tubule, depending on what we need. And we use hormones to help control this. Okay, so there's the overview. Now let's take it apart mechanistically. The point is that, the main point here is that we are focusing on the loops of Henle. Let's see, this is the descending and ascending limb of the loop of Henle, and the vasa recta. And remember, the spaces, the interstitial spaces, are a lot smaller than this. Right? This cartoon highly exaggerates the spaces between these tubules and these capillaries. Remember from lab last week, they were all packed together. There is interstitial space, but it's very small. Okay, so now it gets complicated. This is complicated. The name of this mechanism in the loop of Henle is called a countercurrent multiplier. That's just the name of it. If someone says, describe, someone on a MCAT exam and asks you about the countercurrent multiplier system in the loop of Henle, this is what they're talking about. So let's walk through. This is a figure from your textbook. Let's walk through this together. The fluid entering the loop of Henle, that is to say coming out of the proximal tubule, is isotonic. Okay, let's just remember that fact. The ascend, now let's skip the descending limb just for a moment and go to the ascending limb. Over here. We have active transporters that are pumping sodium and chloride from the tubular fluid, from the urinary fluid, into the interstitial space. And water permeability is low. Okay? Water permeability is low, active transport is high. These arrows, remember that this is an epithelium. So this is including both the sodium-potassium ATPase that pumps the sodium out and the sodium chloride, sodium-potassium 2-chloride cotransporter and the other cotransporters that are bringing the sodium into the cells. So these arrows oversimplify the fact that this is not one membrane, this is two membranes, right? This is an apical membrane and a basolateral membrane. Don't forget that. But the point is, lots of active transport. Lots of active transport from tubular fluid into the interstitial space. So the osmolarity of the fluid goes down as the fluid moves through this process. That makes sense? We're pumping it out. So the osmolarity goes down, and there's the hypotonic fluid at the top of the loop of Henle and in the distal tube. Okay? In the descending limb, so let's now go back. In the descending limb, it starts isotonic, and then it has relatively low salt transport mechanisms. That is to say, not a lot of transporters, and a relatively high water permeability. So let's ignore salt for a moment and focus on water. If this area has pumped salt into here, then the interstitial space is hypertonic relative to what's in here, well, especially here, and water flows from the tubular fluid into the interstitial space because of the hypertonic interstitium. So in the main thing to know, and we'll use this in the next slide, starts isotonic, becomes hypertonic because of water permeability, and because on the ascending limb there was pumping but no water permeability. And then what leaves the loop of Henle is hypotonic. Then, let's skip the distal tubule, it just stays hypotonic. Then as the as the tubule, the collecting tubule moves back down through the same interstitial space, remember there's no separation of space here, they're all bundled together. Water can, if we need to create a hypertonic urine because of water dehydration, this permeability, this water permeability can be controlled, and we can reabsorb water here or not, right? So we can either excrete a dilute urine or a concentrated urine, depending on the water permeability here. Okay, so that's the story. Again, getting into a little bit more mechanism. Let's unpack it a little bit more. Let's see, what did I do? Oh, so the themes. Descending limb, high water permeability, low sodium chloride transport. Ascending limb, low water permeability and high sodium chloride transport that is active, pumping of sodium chloride. Collecting tubule, variable water permeability. Those are the key things. Then in the loop, what's happening here is the loop of Henle is creating this hypertonic interstitium. The next slide is going to unpack that some more. And the collecting tubule has variable water permeability to allow what comes out, which is urine, to either be hypertonic or hypotonic, or isotonic in between. It's the final step that controls the osmolarity of urine. So now so this seems almost magical, right? This idea that this loop can do this. Let's try to understand it. Let's try to unpack this just a little bit. So here's a cartoon that I think helps. Let's walk through it. So let's just start with an imaginary stage, an imaginary step. This never happens because, well, this is imaginary. So let's imagine that we start with an isotonic fluid everywhere in the tubular fluid and in the interstitial space. Let's just start with isotonic everywhere. That's an easy thing to start with. Okay? So now, no flow anywhere. Isotonic everywhere. Okay? Now let's turn on the transport mechanisms in the active transport mechanisms in the ascending limb. Okay? Now remember that this arrow, that this symbol, is active transport. It includes the sodium-potassium ATPase on the basolateral membrane and the sodium-potassium 2-chloride cotransporter on the apical membrane. And it's active transport, pumping sodium from tubular fluid into the interstitial space. Okay? So let's just say that this pump can maintain a certain gradient of, difference of sodium chloride concentration. And in this illustration, it's saying these pumps are strong enough and the membrane is impermeable to water enough to maintain a 200 milliosmolar concentration gradient. Okay? So we've increased the concentration of solute in the interstitial space by 200. We've pumped by 100, sorry. So we've pumped 100 from here, we've pumped 100 out of here and into here. So now by pumping it out, we've decreased this concentration to 200, and by pumping it here, we've increased that from 300 to 400. Okay? So this is all pretty simple so far. And remember that in the descending limb, we have high water permeability, so that in the descending limb, the water simply equilibrates, so that these numbers stay the same. Do you buy it so far? So we've started with isotonic, we've pumped, and then we've equilibrated. Pumped on the ascending limb, equilibrated on the descending limb. And now we have this situation. Okay, we're not there yet, but now let's move a little bit of fluid in the renal tubule. So we're going to introduce two more, so we've brought in 2 more units of the isotonic fluid. These 400s were these 400s before. Some of the 200 left, and these two 200s are now here. Everybody clear? We've just shifted the tubular fluid. We brought in some isotonic. We've shifted the 400. We've let some of the 200s flow out. Everybody clear on what we just did? Because flow is important in this mechanism. Remember that what's really happening is all of this is happening at the same time, but I'm breaking it down into discrete steps in order to try to explain it. Okay, so we've shifted the fluid, and we've allowed this to equilibrate here because it's high, the water's permeability is high, so that these equilibrate. Okay? So already you can see you can see what's happening. Already you can see what's happening. What's going out is hypotonic. In the renal interstitium, we're starting to build a gradient, which is isotonic at the top and hypertonic at the bottom of the loop. The deeper you go into the medulla, the more hypertonic it gets. Right now, even after just one step of pumping and equilibrating and then shifting and equilibrating, we've already seen the theme that we need to build. We haven't gotten all the way there, but these 3 steps show you what's happening. Hypotonic leaving and a gradient in the renal interstitium from isotonic at the top to hypertonic at the deep layers of the medulla, deeper into the loops of Henle. There it is. Now I could walk through this, but the point is, it's doing it all the time. So now if we just take this and we pump some more out, we build that differential again, the 200 milliosmolar differential across, and the arithmetic is going to start to get a little fuzzy, but let's not worry about the arithmetic. The point is, pump solute out of the ascending limb, then equilibrate across the descending limb. I'm sorry. Pump out and equilibrate, then shift, bring in two more small volumes of isotonic fluid, push out 2 more units of hypotonic fluid, and equilibrate. The gradient gets bigger. Do that a few thousand times, because it's happening all at the same time, and we build a gradient that looks like this, with hypotonic fluid leaving, isotonic fluid entering, and a steep gradient of solute in the renal interstitium. So there it is. Okay, so, sorry. This is how the loop of Henle, using this countercurrent multiplier mechanism, which your book tries to simplify into one slide. We've been focusing on this part. This is how the loop of Henle makes the deep medullary interstitium hypertonic. And because this is a highly water-permeable tube, this fluid becomes hypertonic as you go from shallow to deep in the medulla. What's flowing out is hypotonic. We haven't talked about the collecting tubule yet. That's still to come. The point is that this story is trying to make convincing to you this situation. Okay? So now the vasa recta parallels the loop of Henle. This is a slide from your textbook, which I find super confusing because you've got all these arrows and numbers. You don't need to follow the arrows and numbers, even though your mind wants to. The point is, let's just look on the inside. What comes in the vasa recta is isotonic, because these capillaries are leaky to water. Their job is to pick up that water that the kidneys are going to give it. So the blood in the vasa recta is isotonic entering, and it's pretty isotonic leaving. But the vasa recta has a solute concentration that parallels the solute concentration in the renal interstitium. Why did we evolve it this way? It's so that the vasa recta can pick up water. Water is going to be reabsorbed in the vasa recta because we want to recirculate that water back into our bloodstream. We don't want to urinate it away under most circumstances. But with the vasa recta paralleling the loop, it can pick up water without washing out the gradient that the loop of Henle just burned a bunch of ATP in the form of active transport to produce. The loop of Henle burned a lot of ATP to produce this gradient. We want to reabsorb water, but we don't want to wash out the gradient. Just imagine if this was surrounded by peritubular capillaries that were running, that instead of running in parallel, were running, you know, sort of across like they do pretty much everywhere else in the body. Then down here, a capillary would have 300 milliosmoles, and then it would be experiencing this high solute concentration, and water would get, it would dilute. Water would flow out of the capillary and into this interstitium and dilute it. And then it would leave being concentrated, highly concentrated, and it would have washed out this high number here that the loop of Henle spent so much energy to build. So the vasa recta, by paralleling the loop of Henle, allows water to be picked up into capillaries and recycled into the blood without washing out the interstitial solute gradient that the loop of Henle built. All of these numbers are just distracting to me. You might be able to figure out what the book is trying to do with it, but I can't. The point is, water is flowing osmotically. It's not being pumped. What's being pumped is solute, and then water is following the solute. So now, let's look at this data slide, because now we're going to get into the collecting tubule. Okay? So the water permeability of the proximal tubule is high, and it stays high. The water permeability of the ascending limb is low, and it stays low. The water permeability of the the distal tubule is low, and it stays low. So let's just walk through. Proximal tubule water permeability is high, and it's always high. Descending limb, water permeability is high, and it's always high. Ascending limb, water permeability is low, stays low. Distal tubule stays low, is low, stays low. Here, in the collecting tubule, the cortical and the medullary collecting tubule, the water permeability is sometimes low and sometimes high. Let's unpack that. So let's start with a situation where most of you are in right now, where you're fully hydrated, and some of you may have just drank a half a liter of water. Let's walk through this one. And I gave this to you, it's on the second page, or maybe the third page of the handout, so that you can mark it up. Let's walk through this again, because now we're getting to the production of urine. Because we're going to include the distal tube, the collecting tubule. Water enters the loop of Henle isotonic. The loop of Henle builds a gradient, so in this case it's building a relatively modest gradient. 500 milliosmoles per liter in the interstitial space. It produces a dilute fluid leaving. Hypotonic fluid leaves the loop of Henle. And the water permeability of the collecting tubule is low, so that the water permeability, so that the urine has the same osmolarity, more or less, of what entered. So it's producing a hypotonic. Don't worry about the 60 versus 100. That's not a significant difference. It's producing a hypotonic urine. You've just drank a half a liter of water. You don't want to get too diluted. Your kidneys are producing a dilute urine. Water permeability in the collecting tubule is low. So the dilute fluid that was produced by the loop of Henle is excreted. Okay, let's go to the other extreme. You haven't drunk water all day. Somebody told you don't drink water today. Your professor said don't drink water today, and you're a good compliant student, so you didn't. Okay? Your body still is, you've used homeostatic mechanisms to control the osmolarity of your body, and so the blood is still 300 milliosmoles. The loop of Henle built a very steep concentration gradient, going from 300 to 1200 milliosmoles in the deep loop of Henle. The fluid that's coming out is hypotonic, but now the water permeability in the descending limb is very high. This is the key thing. When you're dehydrated, the water permeability, I said descending limb, sorry. The water permeability in the collecting tubule is very high, so that as the collecting tubule goes through this concentrated interstitium, and there's water permeability, water is taken out of the fluid into the renal interstitium, picked up by the vasa recta, and returned to the body. And what the urine that's produced is highly concentrated. So let's tell the story one more time with this slide in mind. Isotonic fluid entering the loop of Henle. Water permeability in the descending limb allows the fluid in the descending limb to equilibrate with the surrounding fluid. Salt pumping in the ascending limb, and this countercurrent multiplier system builds a hypertonic interstitial fluid. This salt pumping produces a hypotonic urinary fluid leaving the loop of Henle. As the fluid flows through the collecting tubule, high water permeability allows the water to come out of the urinary fluid into the highly concentrated interstitium that the loop of Henle spent a lot of energy building. Water comes out, water goes into the vasa recta and is returned to the renal vein. So the main difference, there's two main differences. When you are dehydrated, you build a steeper gradient and you produce a concentrated urine because of high water permeability in the collecting tubule. When you are fully hydrated, which most of you are, you build a less steep gradient and you don't have a permeable collecting tubule so that you produce a hypotonic urine. Okay? Questions? Because now I want to get into the molecular mechanisms of how we control this, because the thing that is controlled is this water permeability. This is what your body detects its osmolarity and controls both the steepness of this gradient and the permeability of this tube. That's how we control whether we produce a dilute or a concentrated urine. But are there questions about this mechanism? It's a complicated story, but I've tried to say it simply and enough times that maybe it's sinking in. John?
Can you go through why there's a smaller gradient when you're more hydrated?
Why there is a smaller gradient, I haven't explained it yet. The short answer is we decrease when we are fully hydrated, there's a hormone, actually the lack of a hormone, that is going to decrease the expression of transporters in the ascending limb of the loop of Henle. When we're dehydrated, there's going to be a hormone that expresses more of these transporters, in the ascending limb of the loop of Henle, so that the countercurrent multiplier is more functional, uses more energy to build more of a gradient. I haven't gotten there, that's where I'm going. Other questions? Hello.
So I was wondering if the osmolarity of the collecting tubule
Okay.
is the same as the vasa recta, then how does water flow from the
It's not the same. So the vasa recta is slightly different. So water is flowing down its concentration gradient. So the concentration in the interstitium, the interstitium is slightly dilute relative to the fluid in the vasa recta. It's the only way it would work. Because water, remember, we're not pumping water. We are allowing water to flow osmotically by using a solute gradient. So the only way water can flow is down its concentration gradient, which means from a low solute concentration to a high solute concentration. Okay, so here is the story of how the collecting tubule controls its water permeability. Okay, so here's the story. The hormone is vasopressin. That is a word that I do want you to know. This book calls it arginine vasopressin because there's two forms of vasopressin. This is vasopressin. It's produced by the posterior pituitary gland. So those of you who have had AMP1 have heard this word before because we talked about the posterior pituitary and we talked about control of water. Now those of you who took that class and were mystified by how are we controlling water, we're talking about its molecular mechanism now. So all those weeks, months between then and now are coming together. Vasopressin is the hormone. So the hypothalamus has osmoreceptors. And the hypothalamus tells the pituitary, I'm either hypertonic or hypotonic. If I'm dehydrated and I become hypertonic, I want, well, first I want to drink. And so the hypothalamus is going to increase a thirst response and cause me to seek water. But it's also going to tell my kidneys, okay, let's conserve water. That's what we're talking about now. We're not talking about the thirst response, we're talking about the water conservation response. A hypertonic hypothalamus is telling the posterior pituitary, release vasopressin. Vasopressin is a hormone. It goes into the bloodstream. It goes everywhere, including the kidneys, and this is its target. Let's get oriented. Here's the tubular lumen. Here's the interstitial space facing the bloodstream. Basolateral membrane, apical membrane. A receptor for vasopressin. Now you're going to tell me the story because it's all here. What G protein is this? GS. How do you know it's GS? It's activating adenylyl cyclase. Doesn't say GS, but we know it's GS. It's activating adenylyl cyclase. This story should start to ring all kinds of bells. Let's spend a few minutes unpacking it. Binding, conformational change of the G proteins, GDP dissociation, GTP binding, alpha GTP activating adenylyl cyclase. I know you're bored with this already. Adenylyl cyclase converts ATP into cyclic AMP. You know all this. And I will ask you to tell me about it once more time, at least one more time, maybe twice, before this course is over. Okay, adenylyl cyclase can be broken down into AMP, that's the breakdown, but cyclic AMP binds to the regulatory subunits of pKa. pKa does two things in this cell. It phosphorylates a transcription factor called Krebs and increases the transcription of specific genes, including a gene for aquaporin. Aquaporin. But you know what that is?
Water channel.
A water channel. The phosphorylation by pKa also phosphorylates aquaporin. The point is, aquaporin containing vesicles in the cells of the collecting tubule, especially the medullary collecting tubule, are now incorporated because we've increased the production and we've increased the exocytosis of these aquaporin containing vesicles into the apical membrane. What does this do to the water permeability of the apical membrane? Increases it dramatically. So this is the cellular mechanism for how a hormone increases the water permeability of the collecting tubule. It's all here. So remember, when you're dehydrated, these arrows of water permeability are due to vasopressin increased expression of aquaporin on the apical membrane. Here's the cellular molecular mechanism. Let's take a break now. And we'll get to urea and overall homeostasis. Now's a good time for a break. You struggled with this as homework, but let me do a quick follow-through on it. So you learned by looking up that the loop diuretics inhibit this transport. By inhibiting the sodium-potassium 2-chloride cotransporter in the ascending limb of the loop of Henle, the loop diuretics inhibit this process directly. They block part of it. They block the sodium-potassium 2-chloride cotransporter, so these arrows are inhibited. So we get more of a situation like this with less of this transport, building up less of an interstitial fluid, so that there is less osmotic driving force. Regardless of vasopressin, there's less osmotic force drawing water out of the fluid, so that the fluid produced is less concentrated. And that's how the loop diuretics work. Now, some of you probably said, well, by inhibiting this solute transport, we make this more salty, and so since this is water permeable, which is sort of true, there's more sodium chloride in the fluid, so more sodium chloride in the excreted fluid, which is also true. So you applied the knowledge that you had before, and you said less sodium transport, more sodium stays in the tubular fluid, more sodium stays in the tubular fluid, less driving force for water to be abstracted from that tubular fluid, production of a dilute urine. And I will give full credit for that explanation. But now you know that by inhibiting this transport, we also inhibit the building of the concentrated interstitium. Let's see, what was the other diuretic that we that I asked you to explain?
It was the one that inhibits Venus.
Oh, a milleride blocks ENaC, so which is not shown here. But the idea here is that in the collecting tubule, let's go back here. In the collecting tubule, by inhibiting the reabsorption of sodium, more sodium is in the fluid, more water is in the fluid, more fluid. That was pretty much what I was looking for the milleride one. The sodium and potassium wasting. So, with the loop diuretics, more sodium, less pumping, more sodium, more of that sodium-potassium exchange. Remember the sodium-potassium channel exchange mechanism? More sodium, more potassium excretion. We have to be careful about excreting too much potassium when we use a loop diuretic. And physicians simply give a potassium supplement, potassium pills, to make sure that the potassium concentration doesn't get too low. Clear? You'll see the answers, my answers to the problems that are on Moodle already. A milleride blocks the sodium channel here, so it blocks the sodium-potassium exchange, and so there is no potassium wasting. That was the point of that question. So, because I didn't explain the loop of Henle when you needed to use it for the problem set, I won't expect you to be able to explain it in the problem set answers. But the other things, and really just showing an effort to try to figure out the mechanism for these drugs, is what I will be looking for. Okay, so we now know the molecular mechanisms for variable water permeability in the collecting tube. Again, low water permeability when we have lots of water and low vasopressin. High water permeability when we need to reabsorb water because we're dehydrated, and high vasopressin. Expression of aquaporin allows water to flow from the urinary fluid into the interstitium. We have a constitutively expressed aquaporin in the basolateral membrane and a variable expression of aquaporin in the apical membrane. Urea. So you know that urine has urea. Does anyone know what is urea from? What is urea from? Why do we make urea? I mean, there's several ways to answer this question. It's a less toxic version of ammonia. That's right. Why do we produce ammonia? What metabolism leads us to produce ammonia? Protein metabolism. A byproduct of eating protein is production of ammonia. Ammonia is toxic, so we invest some energy into detoxifying ammonia and turning it into urea. Fish don't do this. Fish just produce ammonia, and that's because they live in water, and they can, as long as the water is circulating and they get fresh water, they don't have to worry about the ammonia concentration building up to a toxic level. Although if you keep fish in an aquarium, you have to worry about it, because if you don't want your fish to die, you've got to make sure that the ammonia concentration in the tank doesn't get too high. So fish don't detoxify their ammonia. They just excrete it as ammonia and assume that the water that they swim in will get rid of it. We would become, if we didn't turn that ammonia into a less toxic molecule called urea, we would die of ammonia toxicity real soon. Birds do something in between. They produce uric acid, which is that white pasty stuff in their feces. Right? Okay. So we produce urea as a byproduct of protein metabolism. We also use it as a solute in the renal interstitium. So when water is reabsorbed in the collecting tubule, urea actually follows that water and contributes to the renal interstitium. And let's not try to follow all these arrows. The point is that urea contributes to the solute in the renal interstitium. I've used the loop of Henle to explain how sodium is built up in the renal interstitium. That's what this gradient of color is supposed to illustrate. Urea facilitates maintaining that solute gradient. Because if water were to flow, remember, you could have asked me the question, wait a second, if water is flowing out here, then that water is going to dilute this interstitial space. How do we maintain that interstitial space if water is flowing out? And I would have said, well, the loop of Henle keeps building it, and co-reabsorption of urea helps prevent the dilution of the renal interstitium by bringing a solute along with the water. So some of the urea that we produce builds up in the renal interstitium and is used to contribute to that solute gradient. Some of it is recycled back into the body, but we don't need it, it just happens. And when we're fully hydrated, we excrete more urea in order to get rid of it because we don't need that renal interstitium because we don't reabsorb water, so we don't reabsorb urea. Okay? So this is sort of a typical scenario where we're excreting about 15% of the urea that we're producing. We're using the rest of it pretty much in the renal interstitium as a solute. It's a solute besides sodium that we built using the countercurrent multiplier of the loop of Henle. So that's urea. So let's talk about water homeostasis overall. So the loop of Henle and the vasa recta build and maintain a high concentration of solute, mostly sodium. Variable water permeability in the collecting tubule depends on vasopressin. So AVP is arginine vasopressin. It's just vasopressin, and ADH is another name for the same hormone, anti-diuretic hormone. It's the same hormone, just a different name for it. Different books use different language. Vasopressin is the same as anti-diuretic hormone. We've gone through how vasopressin contributes to water permeability in the medullary collecting tubule. And in the absence of vasopressin, low water permeability in the medullary collecting tubule allows water to be excreted. Okay, so those are the, circling back to the broad themes of water homeostasis. Oh, I forgot. We've got a couple of clickers, so let's do some clickers. What is urea? Some of these I've taught, and some of these I haven't taught. I'm curious about your own perception about urea. So the best answer is just A and B. Urea is not the colored compound in urine. Urea is colorless. So urea is not what gives urine its color. And it's not it's odorless. It doesn't give urine its odor either. So urea is a product of protein metabolism, and it's a nitrogen-rich compound. That is the product of protein metabolism. What are the functions of urea? Let me just give you a hint. There's more than one correct answer. What are the functions of urea? Right, A and C. It's not a source of energy. In fact, it takes energy to make urea out of ammonia, and it doesn't bind sodium. Those are just too silly. Okay, so here are, just so you know, here are the chemicals that give urine its color. These are, and I'm not going to test you on this. This is just because it's interesting. Because you were under the impression that urea gave urine its color. Now I'm telling you the way it really is. Does anyone recognize this molecule? Iron in the middle of a ring structure. That's heme. That's heme. These ring structures are heme breakdown products. So the color in your urine is due to the breaking down of the heme from the breakdown and recycling of hemoglobin from the breakdown and recycling of your red blood cells. Your urine gets more colorful, more yellow when you're dehydrated, not because you're producing more of this, but just because it's more concentrated. You've drawn out the water, so the color increases because of the concentration. Okay, so now you can impress your 8-year-old nephews that you know the colors, the chemistry of why their urine is yellow. Just to be clear, this is another breakdown product of heme that is not found in the urine. It's found in the feces, and this is the pigment that gives feces its characteristic color. So it also is a product of heme metabolism. And in fact, the color in bile, the green color in bile that we'll see in the cadaver lab this week, is also a breakdown product. Bile is colorful because of the color of a breakdown product of heme. All of these colors in our body come from the breakdown of heme in hemoglobin. So your blood is red because of heme. Your urine is yellow because of a breakdown of heme. Your feces are brown because of the breakdown of heme. Your bile is green because of a breakdown of heme. They're all coming from heme and hemoglobin breakdown. Okay. So now let's spend the last 10 minutes putting together water homeostasis. Okay? It's important, right? Because if I deprive you of water by locking you in this room and taking away all your water bottles and saying you can't leave until 5 o'clock, your body will adapt for at least 24 hours, maybe 48, producing a very concentrated urine in order to conserve water. If, on the other hand, I give each of you a liter of water and I say you've got to drink it, your body would tend to get diluted, but you'll put a lot of that water into your urine, your urinary bladder, flush it down the toilet, and your body will stay at about 300 milliosmoles per liter. That's a number. Just like pH 7.4 is a number that we control very narrowly, 300 milliosmoles per liter is a number that we control very carefully. So you probably know that if you get dehydrated, like if you're in the desert, you have about 24 hours to find water. If you don't find water in 24 hours or so, you get dehydrated and you die. Have any of you, if you drink too much water, what happens? If you really, really overdose on water, die. There have been cases where runners have been so compulsive about hydrating that they actually went too far. So, I have a lot of slides, but I don't have a lot of time, so I'm going to condense. Remember, if you don't filter it, you can't excrete it. Filtration is the first step of excretion. So, one of the ways that we control water excretion is by controlling filtration. And we've already gone through this. Remember, we have the macula densa in the distal tubule, controlling the afferent arteriolar diameter, controlling filtration in the glomerulus. So control the sympathetic nerves and the feedback mechanisms control filtration. And so I'm going to skip a few slides because they're familiar. I think I reproduced them for you, but this is a repeat of stuff that we went through before. The sympathetic nerves constrict the afferent arteriole. High flow can constrict the afferent arteriole. So we have mechanisms both intrinsic and extrinsic for increasing the constriction of the afferent arteriole. Now I'm going to slow down a minute. Renin, angiotensin, aldosterone. So this is endocrinology, so remember endocrinology is the other AMG class. We focused previously on how angiotensinogen is produced, angiotensin is produced from angiotensin 1, which is produced from angiotensinogen. I'll tell you the story again, but I think you got the idea. Renin cuts angiotensin, I'm telling the story. Renin cuts angiotensinogen to angiotensin 1. Angiotensin converting enzyme converts angiotensin 1 to the functional angiotensin 2. Vasoconstriction. Increase or decrease of glomerular filtration depending, but in this case we're talking about a decrease in glomerular filtration by constricting the afferent arteriole. Okay? Big picture, we control glomerular filtration. If you're fully hydrated, lots of glomerular filtration. If you're dehydrated, let's cut glomerular filtration to the minimum required. Let's skip this slide and get to, this slide is just what your book describes. So here's the story in a picture format. Angiotensinogen, renin cuts angiotensinogen to angiotensin 1. ACE converts angiotensin 1 to angiotensin 2. Angiotensin 2 constricts the afferent arteriole and reduces filtration. Okay, so just a picture way of telling the same story. Let's talk about aldosterone, the last step in the story. So remember, if you're renin, angiotensin, aldosterone. What does aldosterone do? Aldosterone is a hormone, it's a steroid hormone that increases the expression of the sodium-potassium 2-chloride cotransporter where? In the ascending limb of the loop of Henle. So this hormone is a transcription factor, or it activates a transcription factor that increases sodium-potassium 2-chloride cotransporter expression, increases sodium pumping in the ascending limb of the loop of Henle. More of that transporter, more hypertonic medulla. More hypertonic medulla allows us to build a more hypertonic urine. And now, vasopressin increases the water permeability of the collecting tubule, allowing us to use that hypertonic medulla to make a hypertonic urine by controlling water permeability. So the renin-angiotensin-aldosterone system helps us conserve water two ways. by lowering filtration and by helping the loop of Henle build the gradient, and then vasopressin from the posterior pituitary helps us use this gradient to make a concentrated urine by increasing water permeability, increasing water reabsorption from the urinary fluid. Okay, so the rest, did I give you these slides, these flow chart slides? So these are in your textbook, and your text does a pretty good job of explaining it. So let's go through one of these. Just spend the last 5 minutes going through one. So plasma volume. Actually, let me look at the ones. Let's decide which ones I want to go to. Okay, let's do that first. Why would your plasma volume go down? Dehydration? What else could cause your plasma volume to go down? Loss of blood. Right? Hemorrhage, dehydration, there's probably other diarrhea, that's a form of dehydration. So we detect plasma volume in the hypothalamus with baroreceptors and osmoreceptors. And this is not being included here, but the point is, if we detect a low plasma volume, we decrease GFR, increase renin secretion, so now we're getting into the endocrinology. Renin, angiotensin. What does angiotensin do? Constricts the afferent arteriole and decreases glomerular filtration, and stimulates aldosterone. Then, does anyone know, this is endocrinology, but does anyone know where aldosterone comes from? Where in the body do we secrete aldosterone into the blood? Remember, it's a hormone, so it's secreted from a cell into the blood, where in this case the target is the kidney. The adrenal gland. The adrenal gland produces aldosterone. So angiotensin tells the adrenal gland to produce aldosterone. Aldosterone tells the so this is actually a mistake here. Aldosterone tells the ascending limb of the loop of Henle. So fix your hand out. Aldosterone tells the ascending limb of the loop of Henle, pump more sodium, build more gradient, so that we have sodium and water reabsorption.
Can you say that?
Say that again?
Can you what we fix what do we fix on the slide?
Or what we fix on the slide is that aldosterone, its primary target is the ascending limb of the loop of Henle, not the cortical collecting tubule. What hormone primarily targets the cortical collecting tubule in this story? Vasopressin. So aldosterone and vasopressin work together. Aldosterone helps the loop of Henle build the gradient, and then vasopressin helps the collecting tubule use the gradient to reabsorb water. Okay, so here's what we're going to do for the rest of this week. So today's Tuesday. Right? Yeah, today's Tuesday. Thursday, in class, we will have a catch-up and review. I'm pretty caught up. I have a few things that we can use to answer your questions. Thursday in lab, I was going to have a renal experiment, go through a paper, a research paper related to renal function. But I think it will be more interesting if we spend more time in the cadaver lab starting the digestive system. So I'm not going to this is just starting the digestive system in anticipation of next week. So that, I think that'll be more fun, more interesting. So we're gonna work in the cadaver lab, and I will be available to answer your questions about kidney function, anatomy and physiology during dissections of, initial dissections of the cadaver. So we'll just be working on the cadaver, and so stay tuned for more information about that.